everybody. It's a great privilege to be on this stage today. And it's a great privilege to be able to present our work. So I am not a great speaker like the many people who presented here before us. I won't be able to create such a positive wave of energy as Daniel does usually. But I do have a lot of slides. And I will be presenting my work. Before I start, you know that generative AI is now a keyword, right? So everybody is talking about it. But I want to take you a little bit back into the history and talk about San Diego, Hotel Coronado, 2017. Same place, same stage, 6 years ago. Let's get the audio up, OK? And let's get started again. AV team, I gave you a lot of praise. Let's put the audio up. Good area for AI to disrupt. And there are two strategies for AI-driven innovation in pharma to ensure that you get better molecules and much faster approvals. So one is looking for a needle in a haystack, and another one is creating a new needle. So our company developed a pretty comprehensive pipeline that utilizes both of those strategies. We start with a large numbers of molecules, large numbers of hypotheses, and then take it through a process that gives us very high-quality, promising leads that then later we validate and put into partnering and pharmaceutical drug development. But today I'm going to be talking about one technology in AI and deep learning called generative adversarial networks, which is essentially disrupting AI as well. It's a new technology. So yeah, 2017, I'm presenting the first study that we've published in 2016 on utilizing generative AI for a small molecule drug design. And back then, I'm making a promise that we are going to take those drugs into the clinic. And by the way, back then, we were also not as famous. We were trying to fundraise. It was bumpy all the time, as you know, entrepreneurial life. Since then, we raised more than $400 million. And managed to get some drugs in the clinic. So I'm going to tell you about part of this journey, and then I'm going to make another promise. So in the next 6 years, you're going to see if I come 6 years later, we'll be able to show you something cool again. So I need to make all those disclaimers, not investment advice, no forward-looking statements. We're not a public company, but I need to make those statements anyway. What I'm going to show you was enabled by my company. It's a team of over 290 people right now. That's the lawyers allow us to talk about. It's actually much more if you include FTEs and consultants. We do have a platform. So the platform consists of 3 elements. One allows you to identify novel targets. One allows you to generate small molecules. Another one predicts the outcomes of clinical trials. And when I was presenting on stage here in 2017, we already had the platforms. We were ready to productize. Then we launched it. Now you can actually license those platforms and acquire our capabilities, not just 1% of our capabilities, if you license all of them. And currently, 10 out of the top 20 pharma companies have used their tools in one way or another, and many are licensing. But the most important part in drug discovery using AI is to get your own drugs into clinic yourself, without a partner, to demonstrate that you can do it. So now we have over 30 therapeutic programs, and some have progressed into human clinical trials. So that has been enabled not only by myself. So I am just a tech guy with some experience in pharma. And now I'm not just trying to modify the slides to be in the trend of generative AI. As you know, we've been in this field since 2016, since the first papers. But my co-pilot, Dr. Ren Feng, now the co-CEO, is the true magician who managed to do what I'm going to just show you in drug discovery. So he built a huge team of drug hunters that now utilize AI in order to deliver life-saving therapeutics to patients. He has PhD from Harvard, 11 years at GSK, and then he listed a company called Medicilin. It's a huge CRO. Took it to $8 billion, and then joined me after seeing what generative AI can do in chemistry, because we were synthesizing at his company. So for those of you who are not familiar with drug discovery and development, that's a very important slide. Most of you are. Here you see many stages of drug discovery and development with, on the first line, you've got the probability of success. So as you know, it's very low, so only 10% of the drugs that enter human clinical trials succeed. If you're talking about the early stage from the time you identified the target and validated it, very, very few drugs end up in human clinical trials because they fail. And it takes a long time, so second line is number of years, third line is the number of $1 million that it takes for every stage to complete within big pharma companies. But the most important part is target discovery, understanding the mechanism of disease, understanding why disease happens, what's driving the disease, what is the fundamental therapeutic target that you need to either inhibit or activate in order to modulate the disease. And I'm going to tell you the story where we went end-to-end with AI. All the way to phase 1 human clinical data in top line, starting human clinical trials, phase 2 soon. And the probability of success within target discovery and the disease hypothesis is very, very low. So the probability of success is 1 to 5%. So 95, 99% failure rates. And it's also extremely expensive. Most of the time it's done in academia. We're going to show you how it's possible to do it within the industry. So when we started, this slide hasn't changed for many years. We mapped out all the major steps of drug discovery and development, looked at which ones could be automated utilizing AI or augmented, so getting superhuman powers in both AI-discovered targets and AI-generated molecules. And we developed a platform, as I've mentioned, which consists of 3 elements. So one element is Pandaomics. It's a platform that allows you to very rapidly formulate the disease hypothesis, utilizing more than 60 target discovery hypothesis philosophies. So we collaborated with a lot of pharma companies to build it. Chemistry 42 allows you to very rapidly prosecute those targets with your own small molecule drugs. We rely on multi-parameter optimization techniques, and it's not just one global generative model, it's many generative models. And in Clinica, we just recently launched it, allows you to predict phase 2 to phase 3 success probability. And we actually cater with this tool not to pharma, but to hedge funds and banks that bet on small and medium-time biotechs that have phase 2 assets because they are very, very sensitive to success and failure, and people can trade on their stocks. So it's actually one of the ways to validate with your own money. So I'm going to talk only about one program very quickly, one software platform. It's called Pandaomics. It's so easy to use that high school students can use it nowadays, and they do. So we have a paper in review currently with 2 high school students as senior authors on glioblastoma multiforme target discovery and also aging research. So we integrate a very large number of human biological data predominantly from public sources and offer over 20 different AI models representing different target discovery philosophies on biological data. And we also integrate approximately $1.5 to $2 trillion of text data that we get crawling the internet, because again, deep bow to the US government for making a lot of publicly funded data open. So now National Library of Medicine provides enormous databases of freely available data, and we utilize that and many other resources worldwide. And we also track biomedical grants, link them to publications, clinical trials, and other data types. And then we allow AI to do its work on the targets. And then we have multiple filters that allow you to prioritize those targets even from the perspective of commercial tractability. So you can see who else is going after those targets, what other relevant targets are there for your indication, and whether you should be going after this target or not, because it takes 10 years, a lot of money, and maybe it's not going to make a big difference for the patient. So we just introduced today, with the launch of GPT-4, we also have Chat Panda GPT, which allows you to talk to our transformer model. So it now got conversational capabilities. Let's get the volume up. So here we're trying to solve sarcoma,
disease, and gene disease association.
In this case, sorry, psoriasis.
We can start by searching for a gene or
let's say you want to solve psoriasis. You get to the disease page. You see all data types that are being integrated in there. You get the target list, but also you can get a knowledge graph like this. And within this knowledge graph, you can now talk to this knowledge graph. You can ask, okay, what are the most promising targets there? Which ones are going to be toxic? Which ones are going to be novel? Which ones you want to drug with the current drugs? You can either ask the question using already predefined questions, or you can chat and ask Pandaomics about what is the most promising drug for your indication and for a specific patient population. Just wanted to show you that it's not just BS, right? So there was a company called Theranos that never published but made great statements that never worked. And we actually try to be extremely open. And for Pandaomics, we have more than 50 research papers backing it up. Here is the kind of propagation of our parametric algorithms into deep learning and then into generative AI. And one really cool paper, you can find it on www.als.ai, where we found some really fun targets for ALS. We collaborated with Answer ALS Consortium, led by Merit Chutkovits and Jeffrey Rothstein from, she's the head of neurology at Harvard, and Jeffrey is her counterpart at Hopkins, have the largest collection of ALS data. We put it into our Pandaomics, augmented it with real brain samples post-mortem from ALS patients and other public data, found some targets, repurposed some of them, and tested them in the fly model at Mayo Clinic, and many of those targets worked. So actually, it's very difficult to validate targets in different models because models are not representative of human disease, and the best way to test your hypothesis is actually to go human. Because most of the models are not going to be representative of what humans have. And flies are just as good as some of the cell-based models, or even organoids, or even or even mammals. It's very difficult to make a good ALS model. So some of those targets worked, and one of those targets was picked up by one of the startups out of Tsinghua University, and they started human clinical trials. So why I'm showing you this is because we started this project last summer, and human clinical trials started last, investigator-initiated, they started last couple months. So within 9 months, we managed to get a drug in human clinical trials from this study. So as you can see, we don't need additional proprietary data to identify promising targets. But we decided that we are going to automate some of this process. So without automation, we managed to nominate 9 preclinical candidates last year in 2020. In 2021, when we just switched from software to discovery, we nominated just 2 for IPF and for kidney fibrosis. And how many of you here have ever nominated a preclinical candidate? 1? Anybody else? 2? Oh, Mike, great. And how many of you have taken that preclinical candidate into human phase 2 clinical trials? 1, great. 2, great. So big pharmaceutical companies, on average, usually nominate 4 to 5 small molecules using internal R&D. So this is actually a pretty impressive number. You can see we can do it without automation and access to significant repositories of proprietary data. And this is the first case that I know of, and I'm in the industry for a while, where AI discovered the target, generated small molecules, did synthetic route planning. We performed many, many preclinical experiments and many different mouse models, different human-derived models, and then did a small phase 0 human clinical trial. So because the target was new and it was COVID, and I was very worried about taking this into humans right away. And then we did a a phase 1 human clinical trial last year. We started February, ended just this January, and announced that we have top-line data complete. So the product, the molecule was proven to be reasonably safe, and we're planning to start phase 2 human clinical trials soon. So this is just the pipeline that the lawyers allow us to talk about because we have progressed beyond a certain stage. So most of those are in preclinical candidate stage, ready to go clinical. 2 are already in human clinical trials. Out of AI, very, very few AI companies can tell you this story, and I think none can tell you that they have discovered a target, generated molecules, and took it all the way to phase 1 complete. And we thought about, I know that I will have very little time, and Daniel is going to be very strict with me, so I'm going to be accelerating. So I've shown you this workflow that you need to go through if you wanted to start the drug discovery program from scratch and on the market. So we thought about, why don't we try to improve this with robotics for every step of the way, and also go away from not only human error, but also reduce human bias, because most of the heads of the therapeutic area in big pharmaceutical companies, they already know which targets they want to go after, because it's one of the biggest decisions in their career. So they have their favorite target lists. That's why they are the heads of therapeutic areas. So very often they do not go and explore the space around their favorite targets or go way beyond it. And we decided to actually start with target discovery for robotization. And as I've explained, we've got this Panda AI, and Insilico together, they form the basis for Pharma AI Brain, which we already tested on many academics and pharma companies, so got a lot of human feedback, and we tested it in internal pipelines. Now we have an AI layer that runs the lab, and now I'm going to show you what our automated lab can do. So last 2 years, we decided to build our own lab. What I'm going to show you right now is a video from that lab, and also a little bit of a history. It's going to take me 3 minutes, so please don't shut me up. And also, before I start, I want to say that none of those successes that you're going to see there would have been possible without the power of China. So I don't see any people from China here, but many of you have been exposed to a lot of negative news over the past 10 years telling you about how bad China is. It's actually not, and please make it your next destination when you go abroad, because it's open now and you can actually see for yourself. It's going to feel like on a different planet. And they're extremely great in robotics. And they also work very hard. So some of the people worked during the pandemic, coughing their lungs out, and they still made it work. So here is the video. Let's put a little bit of sound on. And this is us, so we're truly global. This is my lab in Shanghai. It's a human lab where we host a lot of drug discovery scientists who supervise the work within contract research organizations, and also supervise the work in the robotics lab. So you can actually control this lab from the office in Shanghai. If you are not familiar with China, 1.5 hours by drive is Suzhou. It's a huge population. It's probably like half of California. They have a BioBay building there. It's also size of Palo Alto, probably. We got a building there with our logo. This is me a year ago presenting the vision that we're going to have. We're going to gut this building and build a fully automated lab. People worked round the clock. 1 minute. Round the clock during the pandemic, so they slept there. During lockdowns, we've got matrices there, and people worked there anyway. Here we have a gas leak. They told me not to come to the work. That's me sleeping. This is today. So I've got a face-activated entrance. That's really footage from December 29th. You walk in. I wanted to look like a spaceship. You see the company history on the left. On the right, you see the dimmed windows. You can undim them and see the robots. And when it's a confidential workflow, you won't be able to see the robots. I have a presentation facility. You've got a miniature version of every room with every robot replicated in 3D. That's me presenting the lab to our partners who flew in from all over the world to see this lab, the pharmaceutical companies that really wanted to see what we do. The real key there is not you're going to see a lot of equipment. The equipment is more or less standard, so you're not going to impress Big Pharma with just the equipment. But the workflow is cool. So especially for those of you who are interested in aging and aging clocks. So you throw in a sample, that's where your job ends. Let's say a patient, part of mouse. And you do the high content screening, so the robot automatically grinds the sample, does quality control microplates, it gives it to CX7 machine and Celigo for imaging. Then part of it goes into the incubator, part of it goes for NGS workflow, so next generation sequencing. So we prepare several libraries, one for whole genome sequencing, RNA-seq, and methylation. We have a few others that we cannot talk about right now. Then the robot picks it up, gives it to Illumina next generation sequencing machine, and you get methylation, transcriptomics, and whole genome sequencing in addition to high resolution and high content imaging. So for every sample, you get a very, very large amount of data. It goes into my AI layer, and here is the secret sauce. So that's where magic happens. AI makes decisions on which targets to go after automatically based on many, many years of human experience. Picks the compounds from a library for those targets where compounds are available. For parallel workflows, you go CRISPR. But here, in this particular workflow, you use real compounds, either from FDA-approved panels or from compound libraries where you have tool compounds. You do some quality control. You microplate those compounds on a liquid handler into into the microplates, and then incubate your sample that you got for the very beginning with those compounds, and incubate it for a certain amount of time at different concentration, and then the entire workflow is repeated. So you get high content imaging, you get high resolution imaging, you get methylation, transcriptomics, you can do aging clocks, look at the aging clock response, multi-model. It all goes back into AI and and checks whether the predictions were confirmed by experimental results. So it learns constantly, 24/7. It also helps us with our current programs, and it generates massive amounts of unique data. So I'll just end with this slide. So now we call it Life Star 1 because it's kind of like Death Star, but for life. So we are making it bigger, introducing new tools, new equipment to get more multimodal data in, and also train those multimodal transformers that allow you for target discovery and chemistry at the same time. But what I want to do within the next 2 years, and again, if I get enough funding and freedom, I'll probably try to do that. We're trying to miniaturize it to 2 rooms, so I can place those target discovery labs with PandaOmics platform into hospitals for personalized medicine. You do need sequencing anyway, but also for personalized drug discovery and also for target discovery. So you could put those labs into places that never even thought about target discovery. So for example, Saudi Arabia or UAE or some other country that has a lot of funding, a lot of highly qualified labor, but doesn't have the history or background in target discovery, because target science is a very unique field. And you could possibly feed those targets into pharma pipelines and enable novel target discovery. So that's it. Just wanted to kind of leave you with one of the last notes. So if you're interested in aging, I co-organize the largest event in aging research for drug discovery. It's about 5 days event, and all the key opinion leaders are there. So would love to see you there too. Thank you very much. If you're interested, just email Petrina. She is the head of AI platforms, and she runs our lab. Thank you so much.
Incredible work what's happened in the last 6 years. Just a very short answer. How is Big Pharma going to compete with you? And what's the future of Big Pharma in this exponential age that you're pulling together?
So we're not trying to compete with Big Pharma. We're trying to collaborate. So big pharma is extremely poor at productivity at early stages, and very often even like a simple management change would kill a lot of very promising programs. So it's very inefficient, very ineffective. And what we are doing, we're trying to bake the cookies, so pre-bake the preclinical candidates or even clinical stage assets to the point where pharma companies can license it. And then they license it and they perform the human clinical studies, or they take later stage human clinical studies off our hands and put it into where they belong, into massive human clinical studies and on the market.
Great. Awesome. Thanks for the great work.
Thank you.
Watch that space. Amazing.